Welcome to the ATP-Bio public webinar series. Thank you for joining us. Before we begin, I have a couple of housekeeping items to go over. Today's webinar will be recorded and posted to our YouTube channel, as are other webinars. We request that you please leave your cameras on, but keep your microphones muted. And then a reminder to please join us on April 7th for our ATP-Bio webinar featuring our ATP-Bio trainees. And now, without further ado, I'd like to introduce our moderator for today, Nicholas Zuhavich. Nick, the floor is yours.
Thanks very much, Ellie. All right. Well, hi, I'm Nick. I'm a doctoral candidate in mechanical engineering at the University of Minnesota under Professor John Bischoff. Within ATP-Bio, I'm a trainee co-lead. I'm working in integrated thrust area 3, cryo supply chain management. I used to be a biological technician working directly for Dr. Hagedorn, and we continue to collaborate on cryopreservation of various coral cell types. Now to introduce our speaker, Dr. Mary Hagedorn received her PhD in marine biology from Scripps Institution of Oceanography and is a senior research scientist at the Smithsonian National Zoo and Conservation Biology Institute. Dr. Hagedorn has worked in aquatic ecosystems around the world, from the Amazon to Africa, has taught many university-level classes, lectures frequently to lay audiences, maintains an active laboratory with graduate students and postdocs, and is a successful researcher and active grant writer. Dr. Hagedorn has created the field of coral cryopreservation and founded biorepositories around the world for coral species. Today, she's the director of the Reef Recovery Initiative, a global coral conservation program. Today, Dr. Hagedorn will talk about the question, can cryorepositories help save Earth's biodiversity? We will put aside a few minutes for Q&A at the end of the talk. During the talk, please feel free to type your questions into the chat in Zoom, and we'll go through them at the end, or you'll be welcome to ask your questions yourselves once we get to that time. And with that, Dr. Hagedorn, please take it away.
Thank you, Nick. It's a pleasure to be here, and thank you, ATP-Bio, and thank you for joining us today. So I'm going to talk about some of the work that myself and my collaborators do in terms of coral, and then bring that forward in terms of how that fits into biorepositories, both for coral and other species. So let me start sharing. Okay. Okay, so as you may know, Earth's biodiversity is having problems, and so I consider many of the ecosystems that we talk about as being on the edge and being somewhat fragile. So we're going to talk about many of those today. But as Nick said, I'm at the Smithsonian, but my laboratory is at the Hawaii Institute of Marine Biology, which you see on the right here of your screen. It's part of the University of Hawaii, and it's in Kaneohe Bay, which is surrounded by coral reefs and patch reefs and all sorts of things. It's one of the best places in the world, really, to work on coral reefs because we're in a large cold soup of Pacific water, and so we have less of an issue with bleaching. We have issues with bleaching, but not quite as extensive as things that are really close to, a continental structure. Okay. But first, I'd like to thank my lab and many of the people who've been in my lab and contributed to this work. You'll see Nick there in the corner or in the center there, and many of the others who've been part of this work. So as I said, Earth's biodiversity is at risk. And the biggest driver is climate change. It's altering our terrestrial and marine ecosystems and warming and acidifying our oceans, threatening all life on our planet. We're seeing more droughts. We're seeing more wildfires, floods. We just had 2 weeks here in Hawaii. The first storm gave us 24 inches of rain. The second storm gave us 30 inches of rain. It's just extraordinary to see this amount of rain come down, which, it seems seems odd. I've never experienced that amount of rain ever in my whole life. And it actually does have a huge impact on the reef, all that fresh water coming on the reef causes it to bleach. Actually, you can get bleaching just from fresh water. So coral reefs are animals. That's the important takeaway. They are not rocks. They make their own skeleton, so they seem like rocks. And they're largely located in the tropics. It's one of the oldest ecosystems on Earth. They support about a quarter of all marine life. That they live on a coral reef at some time. They help produce oxygen. They protect homes and cities, and they create livelihoods, estimated at about 30 billion US dollars annually. Those numbers are kind of hard to come by. So, but first, let's just ask what is biodiversity? Some people don't know what that is. And to me, it's species richness. Think of it as money in your bank if you're a biologist. It's having a lot of species on the planet. And the other thing that people don't often know, a good definition for is genetic diversity. And I like to think of it as having many individuals in a population or gene pool. The more people you have in your population, the less you're going to have genetic problems like inbreeding and other things. So this is sadly what a lot of coral reefs look like today. If you go to the Caribbean in particular, sometimes you don't even see this. You'll see just sand. But there are still places on Earth, such as Raja Ampat, where you have an extraordinary diversity of biodiversity and genetic diversity. I mean, in these places that are still, sort of coral hotspots, you can't even put your finger, on into the reef. There's just coral everywhere. It's extraordinary. But to save diversity, we really need to think about two possible strategies. The first is in situ conservation, where we're protecting and restoring our reefs and natural habitats. But the second really is ex situ conservation. It's really what I'm going to be talking about the most today. And that is maintaining corals and their genetic material outside the ocean. It's an essential assurance, and it may actually, sadly, be the only thing that's left of coral reefs in 100 years from now if we do not change our behaviors. So we are creating biorepositories and using human fertility techniques to help save coral reefs. In ex situ conservation, we're focusing on cryopreservation, and the goal of that is to help maintain cells alive for decades. And basically, you have a cell here, you're starting to freeze it there's some ice crystals forming, and water starts to come out of the cell. And you're using cryoprotectants or CPAs here, which are often modified sugars. They can be a lot of different things, but those can permeate into the cells. There's some that permeate and some that don't. But the goal is to use these cryoprotectants to help stabilize the cell while it's freezing and to prevent lethal ice formation inside the cell. So here's what coral look like, because a lot of what we're going to be talking about is based on coral reproduction, and then we'll get into non-coral reproduction. But the very first part of what we worked on was had to do with creating biorepositories that dealt with gametes of some sort. And here's what corals look like when they are spawning. It's really an amazing event, and it's very, very beautiful. The way you can collect those gametes is if you have an endangered species like a cropper palmata or cervicornis, you can put a net over the coral and you can collect the egg sperm bundles in a 50 ml tube. And here you can see the pink egg sperm bundles at the tip. And another way to do it to collect your gametes is to bring corals into your laboratory or your water system. Put them in. We use floating water flower pots, and we have a colony in each pot. And then you can see this is Claire Lager, my laboratory manager. She's collecting egg sperm bundles from the surface of the flower pot. But this often happens at night, and we use red lights to keep the corals spawning. If you flash them with white light, oftentimes it can stop them. Another way is to build a coral nursery, and this is one of the things that Nick helped with with Claire in Moorea. We built a nursery for some of the species in Moorea, and about a year and a half later, we had the first spawn of those corals on the coral tree nursery. So there's many ways to try and collect coral spawns that might work. But here's again what it looks like. The eggs, sperm bundles are a cluster of eggs and then a packet of sperm inside. Once you leave it for about 15 minutes or so, you have this separation of the white sperm on the bottom and the eggs on the top, and then we collect the sperm. So here's what a coral sperm looks like. It looks very much like other organisms. There are some things that are very interesting and odd, like shark sperm, but this looks like other organisms. And this is what a coral larvae looks like. It's a small one. This is from mushroom coral, and but many coral larvae look like this. Some are larger, some are smaller. Some have symbionts in them, some don't. But they all look about this sort of look like more or less like this. So the first thing that we do is we actually try and assess the sperm, and we use computer-assisted sperm analysis. And this is Mike Henley's work, and he worked on two species in Kaneohe Bay. And after the bleaching event that we had in Kaneohe Bay in 2014 and 2015, we noticed that there was a decrement in many of the species' sperm motility. And here's one of the ones that we noticed, and you can see the tracks for this. It's not terribly motile, those sort of green tracks that are running around. This is another species that's was protected from the damage of bleaching. And here, it's Montipora flabellata. You can see the coral sperm is, somewhere between 80 and 90%, whereas in the Montipora capitata sample, it was probably 20, perhaps, percent motility. So there's differences in species and there's differences in strategies. The one thing that's very, very clear is that when you have a bleaching event, you do get damage to the reproduction of coral. Okay. So we started working on the cryopreservation of coral sperm about 2008, and we started creating a bank in about 2011, 2012. And the benefit of having sperm cryopreserved is you can diversify shrinking populations. And today it's at a relatively advanced stage of development. And the limitation in this practice, though, is that coral spawn, a particular species of coral spawns only about 2 to 3 days a year. And so you're very, very limited in how you can collect your organisms and the sperm. And if you have any kind of event, a weather event or whatever, you can miss a whole year, in which we have done in the past. And so it's difficult in that regard. However, you can use your frozen thawed sperm and you can create new corals. Here's a species of Cryorepositories clethrata that we froze the sperm and we created new babies with it. But you can also use the sperm for assisted gene flow, which we did for Acropora palmata, which is one of the major reef building corals in the Caribbean. We had gone across the Caribbean, freezing sperm from anywhere from 2008 to 2022. And we used that sperm to fertilize fresh eggs in Curacao. So we had Florida sperm, we had Puerto Rico sperm, and we had Curacao sperm all frozen, and then we used it with the fresh eggs. We cannot cryopreserve fresh coral eggs at this moment, although I think people are working on it. And so we did assisted gene flow. It's just a fancy word for selective breeding. And we got amazing results with it. We didn't think it would work. We got amazing results. Unfortunately, these assisted gene flow corals are still in human care. Some of them we have over 600 different genotypes of Cryorepositories palmata that we've created with assisted gene flow. And the sad news is that on the Florida reefs today, from Miami perhaps down to the tip of Key West, there are only about 12 Cryorepositories palmata genotypes left. So in the wild, there's only 12 individuals for this major reef building corals that you used to have to use dynamite if you wanted to, have any kind of a movement of those corals, to build any kind of bay or anything like that. So they were once so prevalent you couldn't get through them. There was a huge thicket. So today we have about 53, 55 species worldwide. Where my colleagues and I have been collecting and banking, and as you can see, the stars show you they're all in mostly tropical areas of the world. This is where we store them. We put them in cryo banks, and this happens to be USDA, but the Smithsonian has one, and Taronga Zoo has a very large cryo bank, and we're also creating more cryo facilities around the world. So about this time, I started working with engineers and teaming up with them. And you see John Bischoff in the center there, and John Daly and Nick and Fritz Kleinhans, Kanav Koshla, a number of people who worked to use engineering practices to help solve conservation issues. And one of the big conservation issue that we had is we wanted to start cryopreserving coral embryos. And this is the mushroom coral, Labactis scutaria. And if you just brought it down to chilling temperatures like 0 degrees, it was very chill sensitive, and so the membranes started falling apart. And you can see that happening on the right here. And this is no cryopreservation, it's just chilling. And so it just made it very difficult to try and cryopreserve them if during that process they were falling apart. And so we knew we needed something much faster, and that's when we decided that we wanted to use laser warming. Now, the conservation benefits for having embryos cryopreserved is that it can help prevent species extinction. And as I said, the coral larvae are chill sensitive. And so enter John Daly and Nick Sahavich, and we started working with the lasers, and were able to thaw the mushroom coral larvae after being vitrified at about 1 million or so degrees per minute, in the millions for sure. And they got really good results that, about 40% of the larvae that they handled and vitrified and warmed and then got to survive was about 40%, and that were swimming after the process, which is actually very, very good. As I mentioned before, climate change is negatively affecting coral reproduction. And so we decided that one of the things that we wanted to do was start working on coral fragments. And we've done a lot of work, a lot of work on coral fragments. And the species that we started working on is this species here, Parietes compressa. And the reason we wanted to start working on coral fragments is that we wanted to get away from reproduction, not abandon it, but have the ability to work on corals 365 days of the year if we wanted, not just 2 days of the year. And we also knew that we could possibly spread this conservation practice more quickly if we could cryopreserve coral fragments. And so here's Parietes compressa. We would cut them into tiny little fragments, and then we put them on plexiglass, and they'd start to grow after a while. You can see they have their symbionts in them, which gives them this brown look. And these are the coral polyps, these roseate structures, the coral polyps. And so working with Matt Paul and other colleagues from UC Berkeley, Brooke Chang, Boris Rybinski, and Mike Henley, we started looking at isochoric freezing of coral fragments. And here you can see the isochoric chamber, and here's a tiny coral fragment in it. And the thing that's unique about this is that it's completely incompressible, right? So you have vitrification solution, which is this blue solution here, no air. You have this very rigid wall, aluminum walls that ostensibly stop any kind of ice crystal growth, because when ice crystals grow, they expand. And so you can imagine, although this is not quite the physics, but you can imagine that as ice crystals start to grow and expand, that the rigidity of the aluminum and the solutions help to sort of push back on that process. So what we found is that we could vitrify them, and they lived for about 24 hours to 48 hours, but they weren't robust. There were still some things wrong. They had some sublethal problems with them, probably with ice formation, that caused them to not thrive. And so we said, okay, we have to take we have to take a different tack. And we started working on anemones, and we started, now this is an anemone here. You can see its tentacles. Now this looks quite large, but it's really quite small. It's only about a centimeter. So they're very tiny, anemones about a thumbnail size or smaller. And we started to try and do a number of things like doing shrink swell, and we wanted to get some biophysical measurements so we could start modeling this because we weren't getting enough answers fast enough. And so we started working on the full anemones, and the same thing was happening. They were just full of water. It was really hard to model them. The shrink swell was just all over the place. And so then Mike got this very brilliant idea. He comes from he was an aquarist at the Smithsonian and worked in coral. He was a coral aquarist before he's got his PhD. And he used to hate aptasia, which this is, and he'd scrape them off the glass. But what would happen is he would notice a few days later that a new anemone would sprout up from where he scraped it off. And so we did this thing called pedal laceration. You see the line here. And we took the bottom part of the anemone and we took biopsy punches and we made tiny little explants. And so that's what you're seeing here in B is an anemone explant. And the nice thing about these explants is that we could work with them quite easily, and they will turn into new little baby anemones in about 1 to 2 weeks. And so we did the shrink swell on this, and you can see this here. And Brooke and Matt and their team did a lot of modeling, and so we're in the process now of coming up with some really good, robust solutions to vitrify the anemones. And so we decided we would try the same thing in terms of hacking the biology of the anemones and to try it with coral. And they're sister groups. They're all in the anthozoans. And so this is a coral polyp. We took a small piece of this coral polyp and we did the same thing. We took some of the tissue and we used a biopsy punch and we created this small coral now explant, which looks, if you look back, looks very much like the anemone explant. And the great thing is now that we've closed the circle, the life cycle, we can take these coral explants that we just have it in one species right now, and we can get them to settle. And so now we have the ability to do the same thing. We can examine shrink swell, and we can make the models for this, and then really try and home in on a non-toxic vitrification solution. The thing that's nice about these explants is we got rid of a lot of the water. Their vis-a-vis is about 60%, which means they're largely solids. And so that makes it much easier for us. And the other nice thing is we can then get the small adult-like structure later. And so we've abandoned a lot of the properties of the adult and sort of reverted the animal to a different kind of sort of developmental adult hybrid. I don't know what you really want to call it, but it's really I think we're very, very excited about this. So I'm going to switch course here a little bit and talk about how we're trying to create a global coral biorepository network. And so I work with this group called the Coral Bio Bank Alliance, which you'll see in a second. And we've been working together for over 4 or 5 years, and we decided to put our ideas about conservation of coral genetic diversity and how to make a biorepository into a paper. And we came up with several ideas, but the main one is that standardization is essential across the globe, and that you can have many kinds of biorepositories. They can be cryopreserved germplasm, they can be tissue and DNA, they can be live coral aquaculture. But the important things about them is that they're targeted. You don't just go out and grab something and put it in a tank under your desk. They're secured. That means you make sure that no one can go in and take things in and out that's not authorized. They're long term, meaning that they're not just for 1 or 2 years, they're for decades. And they're cataloged. They have the thing that's really critical is that you have a robust database and metadata. So here's just visually what some of these might look like. You have corals in aquaculture. You could do cryobanking. You could have genomes and microbiomes, which is we're growing these in our lab. You can have museum vouchers. This is from the Smithsonian. You can have a research. You can have just a research collection, which we have in our lab as well, and do reproduction. And here's the Coral Biorepository Alliance, and they're a group from across the globe. Mostly they're coral experts that are interested in all types of biobanking to try and help stabilize coral ecosystems. And here's kind of where we work. The stars are where all the group is, and they all work in different oceans and do slightly different things. Some focus on just bringing corals in, others cryopreserve. There's a wide variety of us. But the thing that's key in all of this is that the husbandry knowledge should keep many species alive in human care. To induce spawning, you can now induce spawning in human care now, and to rear larvae and outplank corals back into degraded reef is not ancillary. Like we don't go out and just freeze everything because that's not going to help. It's foundational to what we need to do in the future. Because if reefs collapse temporarily in parts, the ability to maintain and reproduce and eventually restore the species is determined by what we know about our husbandry knowledge. So it's a key part of everything that we're doing. And maintaining corals alive in culture is very expensive. So it's the most expensive way you can create a coral biorepository. But for now, we can do this. We know how to keep corals in culture. We know how to create a biorepository with them. And but cryopreservation is going to have to take over because it's millions of dollars. I mean, millions and millions of dollars to maintain small collections of corals in human care. And to have the kind of diversity that we need, cryopreservation is going to be the bulk work of what we do. So here, I'm also working with another group that is part of a CORDUP grant that we have, and we are creating new biorepositories around the world. And so here's Dr. Banazak, Carrie O'Neill, Marhaver, Daly, and me, and we've created 5 large and small biorepositories around the globe, honoring national sovereignty. Now, I think this is really an important idea, is that, the idea of a centralized bank where you're just taking everything and putting it in there may not be the best idea. We really focus on a decentralized bank with some major, banks as well, but you don't want to just go in and just go run roughshod over folks in terms of their biodiversity because they will push back, and so a lot of small Caribbean countries now are doing that. They really are standing up for their sovereign rights, and I think justifiably so. So here's our new Smithsonian biorepository. It's growing, and we just put our first coral larvae, cryopreserved coral larvae in there last week. So let's take a look at reef restoration now and in the future. So when you think about coral conservation in the future and supporting technology versus success over time, the pace of climate change is affecting coral population. We've talked about this, but our ability to capture the biodiversity and genetic diversity is lagging behind that pace. We have live biorepositories and coral biorepositories, which I told you about, and they're successful, but there's very few people, and we're not getting a lot of money in terms of the restoration budgets that are out there. Some of the global restoration budgets are in the billions. I mean, there's a lot of money out there for restoration, and in situ restoration has a lot of people. Its success it's not showing a lot of success right now. It's not because the people aren't good at it or they're not smart. It's because of climate change impacting them. And so they were spending a lot of money and a lot of hope. And it's not working, I think, as much as people hoped it would. And also the other thing that's interesting is that there are many groups, especially that want to do in situ restoration, that are not. They're also risk averse in terms of wanting to use the cryopreserved material that we might have. In fact, as I said, The assisted gene flow corals are still in human care. We've had them since 2018, and we know that their genetic characteristics are better than the parentals of, say, Florida, but still management has not really caught up. And so you have this tension between science and what science can do and sort of management and policy and sort of just the lay public. And so there is this lag, and I think there is some risk aversion going on, and hoping that will change in the future. So, but the future must embrace both. You must have live biorepositories, coral biorepositories, cryorepositories, and do in situ restoration so they can go back and forth and feed each other. You shouldn't do one over the other. And I think giving more focus and money for the ex situ biorepositories would really help the in situ restoration processes because you would have biosecurity. So, in terms of, what's happening around the globe, this is, Florida in 2023 and 2024, we had this massive heating event, and it basically extirpated and made a Cropera palmata and a Cropera cervicornis, the main reef building corals in Florida, functionally extinct. And so, if the world were to dramatically reduce its emissions tomorrow, however, the oceans will continue to warm and acidify for generations. Coral reefs and many other ecosystems must somehow survive through this long climatic reckoning for sure. So we must think beyond this. We must think beyond just what we can do on Earth, or what we can do in terms of trying to help coral reefs. And we must have ultra secure biorepositories. These will be key, and there are a number of them on Earth. And the idea of some of some of the ideas I'm talking about really parallels what we did with supercomputers. In the 60s, people knew that they could build supercomputers, and they started working on some of the ideas behind them, but they knew they didn't have the technology. But they knew that the technology would catch up to this great idea. And so we're hoping that will happen too with ultra secure biorepositories. And the one that we're going to tell you about is a passive biorepository on the moon, where space will be protecting Earth's critical species and ecosystems. It's going to take us decades to build this, but it will hopefully span centuries in terms of its biosecurity. And in terms of our group, we have a lunar biorepository group that has again been meeting for many years. I think COVID spawned a lot of these groups. So you can see we have marine and terrestrial reproductive biologists, Smithsonian museum experts, ethicists, ATP-Bio systematists, ecologists, and National Ecological Observatory Network, wildlife, medical, and engineering cryobiologists. We have, when we think about the moon and space, we have Smithsonian Astrophysical Observatory engineers. We have lunar robotics, lunar geologists, and space communication and engagement. So we have a wide variety of people on the group, but it will certainly have to expand as we start to move out into space. As I mentioned, biorepositories can help maintain the biodiversity or the genomic web of life. But they're susceptible to natural and man-made disasters. And here's Hurricane Melissa in Jamaica. And many of our biorepositories mirror human populations, which live in large corridors along our coastlines. And so this is where a lot of hurricanes and other storms come in. And so they certainly are at risk being in those areas. But our inspiration in thinking about a lunar biorepository was the Svalbard Global Seed Vault, which is a passive biorepository in Norway that is about 500 feet under the Arctic Circle. It's an old mine, and they use very little energy to maintain seeds of all types around the world at about minus 18 degrees centigrade. But what we wanted to do is we wanted to think about not only just seeds, but animals and other kinds of organisms as well, which must be maintained at a much colder temperature, minus 196 degrees centigrade. However, there's no place on Earth that's cold enough to support a passive cryopreserved wildlife biorepository. So we really did have to think about looking towards space or the moon for cryogenic storage. And the other challenge that we had was that the cryopreservation of sperm and oocytes for most wildlife species is largely unknown. I mean, people think that we can do this with all this different wildlife, but it's mostly sperm that's cryopreserved. There's some embryos and oocytes now, but it's not really that well documented. And so we opted to go with a different kind of cell. Opted to go with cryopreserved skin cells that have fibroblasts in them. And through transformation, we can take those cryopreserved fibroblast cells, we can thaw them, we can expand them, and we can transform them into eggs and sperm for some species now, and through species recovery using surrogates for some organisms. So in terms of what we want to do before we even, think about going to the moon, we want to do some pre-launch testing and assessments, especially in terms of the sensitivity of the cryopreserved fibroblast cells and other cells that may be part of this collection to radiation equal to lunar transit or waiting on the moon or whatever the case may be. And to do this, we will have to look at DNA integrity, morphology, growth, differentiation, and the ability to transfer. There's many, many things that we will look at to look at the impact of radiation on the cryopreserved cells. But in addition, we're going to use the tardigrades, and one protein that we'll use is the damage suppressor protein, which will help hopefully help reduce the effect of radiation on the samples. There are many other tardigrade proteins and other things that we can also include. Tardigrades can generally withstand 4,000 times the amount of radiation that a mammalian cell can withstand. So, in terms of what we already know and what we already have, we have a conceptual design, which you see here on the right. And basically, it's a series of boxes, and one has a cryocooler. This is a small Stirling cryocooler, which will keep the samples at a cryopreserved temperature from liftoff to landing on the moon. And if they're coming back from landing on the moon to landing or lifting off from the moon to landing on Earth. And then here's where the samples sit, and there's going to be a wide variety of sensors that will measure radiation and temperature and other things. But when you go to the moon, you don't go in a straight line. You actually go around the Earth, you come through, and then you pass by the sun and you go around the moon a couple of times. So temperature stability during the transit is actually very important because it takes about, I think, 3 days to get to the moon or so. And so you really have to make sure you have good temperature and radiation stability in what our conceptual box or cryofreezer will be. And we also have some bags that we'll probably use from Instant Systems, and these are sterile cryolock bags that the fibroblast cells will go into. Some will be transformed by different kinds of maybe antioxidants or, as I said, tardigrade proteins. We'll have a variety of different things that we will use. We will more than likely have a parallel or near parallel biorepository on Earth, so we can compare how well things once they get to the moon are doing, and if there's problems. We don't want to just put things there and not think about that they're being damaged and we're just not thinking about it. So another thing that we came up with was what language do you put on a bag? We're speaking English today, but if it's going to last for 200 years or 500 years or 1,000 years, will English be that language? Do we put a DNA code on? Do we put pictures so how we label these will be interesting, and how we care for them will be really interesting. The goal is to store species to help or maintain Earth's ecosystems, or could be used in extended space travel and for terraformation. And scientists and professionals from around the world will be aided by AI to help drop prioritized lists. Likely we'll start with U.S. species, which have about 150,000 species that we could start with. There's a lot of issues with the Nagoya Protocol, which I'll touch on in a second. We also want to have about 100 to 200 individuals, if we can, per species. That's a tall order. I'm just going to quickly go through these slides. They're just really beautiful pictures, but we'll have forests, species, Arctic species, grasslands and savannas, coral reefs, wetlands, alpine regions, desert and related species, deep sea biomes, extreme environments, food webs. This will be very, very important. All sorts of food webs, agricultural and aquacultural species, species that build and maintain soils, microbial and fungal webs, plants that must be cryopreserved as the critical ones here, which are chocolate and coffee. When we think about the animals that we might want to think about, we'll want top predators, herbivores, insectivores, pollinators, ecological engineers, just as a beginning brush, and there's going to be many more additions, I'm sure, besides this small list. So this is on the right here is the south pole of the moon. And these blue dots are these things called permanently shadowed regions. What that means is that they get very little sunlight and they're in shadow. And many of them have year-round cryogenic temperatures. As long as you don't get a lot of solar energy, you have temperatures around cryogenic temperatures or even lower. And this is probably where we will put the biorepository. It's also where a lot of the lunar bases will be because in these permanently shadowed regions, there is water. And water can be used for fuel and for water. And some of those permanently shadowed regions will be hotly contested, I'm sure. The cryo samples will fly in unmanned missions using rovers to build and maintain the biorepository. You can see here's a unmanned mission. Here's the rover that's going to pick up the samples. And this is just a conceptual design. I mean, this is a PS, permanently shadowed regions. They're not going to be nuts on the surface, but it kind of just gives you an idea that they're, going to be in a permanently shadowed region. We'll more than likely put the underground, and the rovers will go underground. And how we'll do this, whether we'll dig with an auger, or whether we'll be in caves, or whether we'll build something, we're really not sure at this moment. But you need to have enough meters of moon rock above you to block out radiation, and you need about 8 meters to have no long-term impacts on the samples. And that's what we want to make sure we don't have. We don't want to have the samples sitting there getting destroyed by radiation. And we'll more than likely have robots or some kind of specialized rovers within the biorepository to stock it if we have some kind of cave or cavern. If we decide to just, put them in the ground like a plant, if you will, where we till and then plant it, then the rover would do that. Some of the PSRs, as I said, contain frozen water, and its crystalline structure may reveal the moon's origin. So many NASA scientists are very interested in bringing that water back to the U.S. and studying its structure. So what we have in terms of the cryobox, we can bring those samples back in a cryopreserved condition so they can look at the crystalline structure. So the onboard cryofreezers will do that. And so these cryofreezers will probably have a dual process. They will bring samples to the moon, and they will bring samples of frozen rocks or frozen ice or whatever it might be back from the moon. So some of the technologies, not much of it, some of the technology for these things already exists. And SpaceX says they're going to start launching starships, which you can see here. You can see a hangar here. So there's the size of the starship is really huge. And they expect to start launching in 2028, and they can carry 150 metric tons. And although I think it's wildly hopeful, they said they are going to be launching 10,000 of these starships in 2028, a year. So we'll see. Even if they launch a couple of them, I think that would be very good. Now, perhaps one of the most important and the most challenging part of this whole thing will not necessarily be the technology, but it will be building robust permitting, partnerships, and ethics and governance for the biorepository, because it will be complex. Permitting, we need to collect globally, and we need to consider the Nagoya Protocol, and that will take time. We spoke with the Earth Biogenome, which collects DNA globally. They have five lawyers that they keep on staff just to deal with Nagoya and permitting through Nagoya. Ethics. Some indigenous groups consider the moon sacred. Communication and trust building will be critical. Governance, such as who owns the PSRs, how do we ask for the land, how do we keep the biorepository safe from geopolitical conflicts. There'll be a lot of issues in terms of governance and partnerships. We need to expand our partnerships. We now have starting partnerships with Earth Biogenome Project and Rhodium Scientific, among others. So in summary, the Lunar Biorepository focuses on global catastrophic risk reduction for Earth's biodiversity. It will foster global partnerships and diplomacy. It will inspire hope and may be one of the most important collections ever made for the good of all mankind. So thank you to ATP-Bio and our funders, and I'll take any questions if there are any. Thank you.
Wonderful. Thank you very much, Dr. Hagedorn. First, we'll look at a couple of questions that have been posted in the chat, and then we can open the floor. So first, 53 species currently banked. How many species are left to be preserved? I imagine thousands.
Yeah, it's a really good question too, because, I think naively when I first started this, I thought, oh, we'll save all coral species, it'll be perfect, But there are, that we know of, there are well over 1,000 coral species worldwide, and there are many that we don't know of. But many oceans are going to have to prioritize their species. And because we can't save everything and do the kind of job that we would like to do. And so if we can prioritize species, now the Great Barrier Reef has done this. They have approximately 400 species on the Great Barrier Reef, and they have prioritized it based on what they could keep in human care, so what they can take care of and settle and grow and have reproduce, and what and the function that they serve on the reef. So they're doing it based on function, and they got it down to 20 or 25 species. So I think many oceans and folks in different oceans will have to go through the same prioritization, and that's really an important thing before we even start a lot of these cryobanks, is to have the species prioritized. So we know what we're going after, and we can do a good job, and we can get enough genetic diversity in those species.
Thank you. So is the biopsy punch method considered clonal reproduction?
Oh, absolutely. All of it's clonal reproduction. In fact, we've gone from the biopsy punch. We're now using a blender, if you can imagine. And it works really well because the biopsy punch took us forever. So we can use a very sharp blender and just pulse the coral, and we get thousands of explants within just a few minutes. So yes, it's all clonal. And the good thing about doing the clonal stuff is that you know how many genotypes you have, and we have the ability to reproduce the corals in human care. And so you can then create more diversity from the amount of clones or individuals that you have. I hope that answered that question.
Oh, sorry. I think we have a question. Sorry, Dr. Capron.
No, that was my question. And my second question was also just a matter of terminology, Mary. Is terraformation the creation of an ecosystem on another planet? Is that what that means?
Yes, it is. And so, NASA has a short list of organisms that are essential for terraformation. And we'll hopefully, have some kind of meeting in the near future that focuses on some of those organisms, and see if there's more see if there needs to be expansion on that. The sad thing is that some of the organisms that are needed for terraformation, we really don't know how to cryopreserve that well. And so there needs to be a lot of work done on some organisms. I mean, insects in particular, microbes, even though we can freeze microbes in the lab, we're mostly freezing them for their DNA. To grow them back up and have them prosper really we don't know how to do that well. And so there's a lot of work that needs to be done on cryopreservation and some of the most fundamental organisms that we need for life on Earth and other planets as well.
And is one of the organisms that we're going to need people like you? I mean, no, I mean, seriously, we have these robots up there. They've preserved all these things. Somebody launches to Mars because Earth has become uninhabitable, but the only people going to Mars are oligarchs or something, and they don't know any biology, and they don't know what to do with this stuff.
Yeah, definitely you're going to need people who know how to thaw and grow. So that's why I made such a point of like for the coral husbandry, you've got to have people who know how to grow these things out, who can thaw them, who can make them grow, and that is critical. So our growers, our farmers, our people who really know the biology, who know about biological systems are going to be critical as well. I should probably put them on the list.
And I guess one question further to that is, at some point, if an attempt at terraformation is made, are you already designing a process for deciding what you're going to use? And are you going to want to hold back a portion of whatever you use because maybe that environment won't work, but 100 years later, with new advances, terraformation could be on an extra solar system planet.
Right. You're asking a very important question about biorepositories. And when people want to build them, you work backwards and you answer that question first. How many do I want, right? How many species, how many samples? You really have to project yourself, 10 years, 100 years, in this case, 1,000 years into the future. And so that kind of thinking is really part of this as well. I do think that we will be working in space. It's coming, and hopefully there will be laboratories and things like that on the moon. And all of these samples can be, be, you know, thawed and perhaps regrown. Obviously the fibroblasts can be re-expanded, seeds can be regrown. Same thing with, algae and other organisms. So there will be the opportunity to be able to expand them, and we will hopefully have a parallel bank here on Earth. Answering those questions, I think is, fundamental questions of how much we need is really, really important. And I've talked a lot with Harvey Blackburn at USDA, who keeps all the germplasm for our agricultural species. And he said, take how many you think you're going to need, and double it and double it and double it. So it's really an important issue, and having as much material as possible is really a good thing.
Thank you.
All right. We have a question. Is the problem of preserving coral also a problem of preserving coral plus symbionts? And what about making coral or their symbionts less sensitive to bleaching events? Is that something people are working on?
I think that's something you should answer, Nick.
Oh no. All right. Well, I'll hit it really briefly because this is not my show. But yes, 100%. It is a question of the coral and the symbionts. There is at least one group that has reported in the literature success preserving both coral larvae and the symbionts together. But I don't know whether that has been replicated. My experience, and I think the experience of a lot of our collaborators, is that the symbiotic algae that live inside the coral and the coral system itself, they are cryobiologically very different. They have different permeabilities, they have different water content and so forth, different sensitivity to cryoprotectants. So right now, the avenue that I'm working on and Mary's working on and our collaborators generally is we're thinking about these as two separate systems: cryopreserve the symbionts, cryopreserve the larvae, bring them back together in the end. But it'd be fantastic if we can reliably and across species get it all cryopreserved together. I don't think we're quite there yet, but
In terms of the other part of that, making them less sensitive to to warming and bleaching. The There are people who are working on that, right? They're doing CRISPR. They're doing CRISPR on that, and I think it's really important because we need to have short-term fixes for restoration because that's what's happening. I mean, a lot of the corals are dying because of warming events and they're bleaching and just dying. But I think at some point we have to recognize that this is a dual edged sword of warming, which causes bleaching, but acidification. And really, as much as you'd like to think that you might be able to CRISPR your way out of anything, you cannot CRISPR your way out of the chemistry of the ocean and the change in the chemistry of the ocean because as we get to higher and higher parts per million of CO2 in our atmosphere, it forces more CO2 into our water, causing a lowering and lowering of our pH. And when it gets to something like 550 or something like that, corals will stop growing. When it gets to like 600, they start dissolving. And it's not far from now. It's really not far from now. And it's scary because we don't have any fixes for that other than biorepositories. And so that's why I think it's so critical that we start thinking about this and not stopping and giving up hope that we can help restore and help, create some fixes for our coral or any other species as a matter of fact, but that we actually are working hand in hand with biorepositories.
All right, thank you very much. So Dr. Fahey, of course, has many questions. And
Right.
so we'll try to get to one or two. We're coming close to time here. If disaster struck on Earth, would the robots on the moon be able to continue? And if not, would the repository stay safe?
That's a good question. The robots will be hopefully solar powered, and so as long as one of the big issues on the moon is fluff. And fluff is the surface of the moon that's about 3 or 4 centimeters. And I'm sure you've seen those famous pictures of the astronauts' footprints on the moon. That's fluff. And it gets into everything. And so my concern about the robots is not will they run out of power or anything like that. It's actually the dust on the moon getting into their gears and just grinding them to a halt. So keeping things clean is going to be really important. And if that becomes, we're having a lunar biorepository meeting at Smithsonian Astrophysical Observatory in 2 weeks, and that's actually one of the questions we're going to ask. We're going to try and project into the future what biorepository schema is going to give us the best results. And so, yay, we have this wonderful robot that can work. But over time, if it grinds to a halt and it can no longer work, or other robots can't, it may be better if we just use an auger and plant the samples in the ground below the surface, like 8 meters below the surface. And then the problem is coming back. Can we find them again? How do we locate them? What language do we use? And I keep thinking about the Egyptians and the pharaohs. I mean, the Egyptians did a darn good job of making biorepositories. They've lasted 3 or 4,000 years, and I can only hope that we could do an equal job to that.
Wonderful. Well, we're up on time, so I think we'll close here. Don't miss our next ATP-Bio webinar, April 7th, with ATP-Bio trainees. Please look out for that invitation coming up. And I'd like to thank Dr. Hagedorn for taking this time out of her day to share her ideas with us. Thank you to Ellie for coordinating, and thank you all very much for attending, and we'll see you next time.